Okay, so I'm going to go over this part that never got recorded. You've already known what an allele is. It's a different version of a gene. So some genes only have one allele. One allele has been swept to fixation, and any change would be perhaps deleterious. So in other cases, there can be hundreds of different alleles for the same gene. So instead of saying version, we'll say that we get one allele from mom and one allele from dad. So which of the patterns can't belong to a child of this couple? 1 second here. So in this one, of course, unless you end up with a haploid child, C and D are both impossible. Now, what kind of cell is this? A, B, C. Now, I want you to remember, okay, so a prokaryotic cell, for now, remember it doesn't have a nucleus, but there are bacteria that don't have nuclei, and there are archaea, which is the other big domain that doesn't have a nuclei either, or any other organelle. So, if you look at this cell, it's pretty obvious that we've got a eukaryotic cell with a nucleus, mitochondria, smooth endoplasmic reticulum, rough endoplasmic reticulum, Golgi apparatus, a lysosome that breaks things down, microfilament, all sorts of different things, and ribosomes that translate the mRNA into actual proteins or amino acids and then proteins. So, who are these guys? Darwin and Wallace, Watson and Crick, Macklemore and Lewis. If you notice, what they have is they have a model of a molecule, and it's actually a model of DNA. And they built a model, like with Legos, essentially, right? And they figured out how to, they figured out the actual molecular, the way, excuse me. Okay, I believe I'm overwriting this. Okay, so who are these guys? Wallace, Watson and Crick, Macklemore and Lewis. These are Watson and Crick. They're the ones who actually discovered DNA, or the structure of DNA. And here they actually have a model that they built. They also had, they were fortunate in that there was this woman named Franklin who was an X-ray crystallographer. And they got a hold of her data, and the data actually gave them the insight into what the actual structure looked like. So there was a point at one point where it was thought that it was not a double helix, but let's say a triple helix. But then they got the X-ray crystallography, and they realized it was a double helix. There were a number of other people, it turns out, working on DNA at the time. One of them was Linus Pauling, who had already won a Nobel Prize in chemistry. But there was a conference in London, and that's in fact where Watson and Crick got to look at the X-ray crystallography. Well, Pauling couldn't go. He was put on the watch list of the United States government as a communist or something like that. So if he had gone, he actually might have won another Nobel Prize. But doesn't really matter. These guys did it. And after that, kind of biology was really changed. But it didn't really change. No, I guess late 70s. So, what is a complementary sequence to these base pairs? So, you have cytosine, guanine, thymine, adenosine. Now, I don't know if you remember this, but a C always goes with a G. So, in this case, if you start on the left, okay, C, what's the complement to that? Well, it's got to be a G. A G must be a C. Another G must be a C. A T must be an A. And an A must be a T. Okay, so here we have, let's see. So, okay, so here we have the DNA up here. It begins to unwind, and it's going to make a copy of itself. And so you have this strand, and so here's a parental strand right here, and right here, right? And if we follow it up, well, these are the actual new strands right here, the ones in blue. And they're the complement to the base on the parental strand. And so, since those bases go together in that specific way, C and G and A and T, there's a pretty great deal of fidelity in the copying of it. So this kind of thing goes on when a cell is dividing by mitosis. Cells do that all the time. And then meiosis, the same thing happens, but it's a bit more involved because you're going to end up actually with 4 haploid gamete cells. So here's another review. An investigator finds a small amount of tissue at a crime scene. What will a forensic scientist perform to amplify the small amount of DNA in the sample? So remember, now it turns out in the past you needed a lot more DNA because the mechanism that you use to actually amplify or replicate the bit of DNA that you want, the section of base pairs, you needed a lot more. But now we have these machines where all you need is a little tiny amount, and then you use something called PCR. So short tandem repeats are they're used actually in a lot of different things, but certainly DNA fingerprinting for one. So in forensics, these STRs, in this case we have a tetra repeat, TAGC, a whole bunch of them. And then on either side we have what are called primers. There are some universal primers that kind of all organisms share. And then you can also make your own primers for the specific sequence that you actually want. And so a repeat can be what? Can be, let's say, 3 of these repeats. It could be 100 of these repeats. It could be 1,000 of these repeats. Now, the thing that you have to remember is that there are DNA fragments of different sizes. So, okay, so here are these fragments of, let's say, tandem repeats. What you do is you take these different samples from different individuals, let's say, and you put a sample in each of these little wells that you see up here. You use a pipette to do it. And then, depending on how big the strand is, so a small fragment will travel, you then apply an electric charge from negative to positive. The fragments start going down, as you can see. And the the little fragments go fast. So, right here, the large fragments move much more slowly. And you get all these bands, and then you compare those bands to, let's say, a rape. Or you have DNA that you've recovered from a crime scene, and there's a database, and you compare all these different bands, there are usually about 13 of them, and to see if you get a match. So, oh God, come on, stop that. Okay, so a chromosome is basically one long DNA molecule, millions of base pairs long. In fact, the whole genome is about 3 trillion base pairs long. So, how is it that this one continuous piece of DNA is actually organized into discrete genes? Well, okay. A gene is a stretch of DNA that encodes for a particular protein. Because actually what it's coding for, each 3 bases, a triplet pair, let's say you had, let's see, C, G, C, that's a codon, and that will code for exactly 1 amino acid. 1 amino acid, okay? And then, if you string a whole bunch of amino acids together, excuse me, you actually get a protein. So you have a stretch of DNA that codes for a particular protein. So a gene really doesn't do anything. What it is the instructions for making a protein. Proteins are the ones that actually do the work. So here we are. How do genes encode for proteins? Well, they, as I mentioned, there's these 3 base pairs, C, G, C, in this case, let's say, and each one of those codons, depending on the order of the bases, will code for an amino acid. There are 20 amino acids, there's actually more, but in living things there are only 20. So, here we are. Now, now we're getting into something called transcription. So here we have DNA, and this is inside the nucleus, okay? Inside the nucleus. And what happens is the DNA unwinds, and you get a complementary RNA made from the DNA. Now, the only difference between an RNA and a DNA is they have the same complementary bases, except that a T is, I believe, yeah, a T is turned into a U instead of an A. But doesn't matter. So you get what's called a messenger RNA. The messenger RNA is then ferried out of the nucleus and into the cytoplasm. And there's this nuclear pore right here. And then we've got this mRNA floating around. And the mRNA is then read by a ribosome. And that ribosome then codes for, it's actually tRNA at that point, and it codes for different amino acids, and here we are. So each of these are little amino acids here. And then if you string them all together, you get a polypeptide. In other words, many, many amino acids strung together, and that's how you get a protein. So this is the actual sequence of how you encode a protein. It only goes in one direction. It goes from the DNA, you get a transcript, which is the mRNA. The mRNA is ferried out of the nucleus and into the cytoplasm, and then it's translated by a ribosome, which is also an RNA, and that is translated into amino acids, polypeptide, and an actual protein. You cannot go from a protein back up to a stretch of DNA. Doesn't done. In fact, this is called the central dogma. So, here we have DNA transcription. Here's the gene to the mRNA, the transcript, translation, protein made of amino acids. So, transcribe, that's to write, or type, or print out. You don't even have to know what it means. All you have to know is that this is the order that it's written in. And the transfer from one place to another. So in this case, you have the messenger RNA, and it's transferred from the nucleus. It's first transcribed into mRNA from the DNA. It's then, the mRNA is then transferred out into the cytoplasm, where it's actually then translated. So, translation, to turn from one language into another. So, in this case, we're taking a whole bunch of base pairs, triplet codons. Each one codes for a different amino acid, and they're strung together. So what you're doing is you're translating from, in this case, RNA, which was a copy of the DNA, into amino acids and a protein. Now, it should be noted, in each gene, in each cell, you have a full complement of all the genes in the human genome. I said there's about 3 trillion base pairs. There's actually only a total of about 20,000 genes. And there's a lot of other parts of the genome that don't actually code for proteins. And it used to be thought that this was all junk DNA, but things have actually changed. But anyway, we'll start with here. each cell has a full genome inside of it. The thing is, there are some genes that are expressed in all cells, and then there are some genes that are only expressed in certain cells. And the question is, how do you make sure that the ones that are expressed in certain cells are the ones you want, and not a whole bunch of other genes? Well, part of the deal is that the DNA, it's one long strand, right? It depends a lot on how it's wrapped around what's called a histone protein. If it's tightly wrapped, it cannot be transcribed and translated. If it's loosely wrapped, then it means it often can be. Now, there are some genes that are only used at certain times in a life cycle. So during development, there's these Hox genes that are turned on and off. And, well, they're not exactly turned on and off, but we'll get to that in a minute. But there's these Hox genes, and as an organism, whether it's a human or anything else, goes through development until it's a mature adult, these Hox genes are they're turned on and off. And after that, they're not used anymore. And so they're actually silenced after that with, they attach what's called a methyl group, but you don't really have to know that. But anyway, in this slide, what we have here is on the left, we have muscle cells. On the right, we have nerve cells. They look pretty different. A lot of them actually use a lot of the same genes because you have genes that are part of, let's say, turning glucose into ATP, right? So that's going to be all genes, all cells. Some cells need more oxygen than others, and so the muscle cell obviously is going to have more of the enzyme that breaks down glucose and turns it into ATP. Okay. So, gene expression, here we are again. All of your cells have the same exact instructions as I mentioned. This is the manual, right? And then genes expressed in muscle cells will say you have a build, a strong, a heart, a muscle. Okay. Now, genes expressed in an eye cell. You have a build, a dark, a brown, eyes. Now, I just want to make sure something though. Genes don't exactly code for a specific trait like a muscle cell or an eye cell. They code for a protein or a bunch of proteins, all made out of amino acids. So, the thing is, you have to get the gene that you want made, or the protein that you want made, you have to get it to be expressed properly. So, both parents give a complete instruction manual to their offspring. From the mother, we have from the father, we have the sperm, got to have blue. Let's see. For the egg, we have pink. Now, it turns out the egg has 23 chromosomes. The sperm has the same exact 23 chromosomes, but in the egg there's only one copy, in the sperm there's only one copy. And then when there's fertilization, you create a zygote which has 46. And that means you have 2 copies of each gene. They're called homologous chromosomes. Okay? So a zygote has 46 pages, or basically 46 chromosomes. There's actually 2 of each of the 23 chromosomes. So here we have a muscle cell. Gene A, gene B, gene C, gene D. There's all these other places in between that don't code for anything. Well, they don't exactly do that, but for now, we'll say that. We get a transcription of A into RNA, and then it's translated into protein A. We also, for this muscle cell, we get transcription of this RNA, translation into this protein, and then transcription of C, and we get protein C. And gene D is not used. Okay? And so it's silenced. We have a neuron. We also get translation into protein A. Gene is off. Gene B is off. C is translated. And D is translated. Now, I want you to notice something. Okay, so let's see here. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Okay, so it turns out protein A is the same size, right? But it turns out that in gene C, there's actually, let's see, 5, 10, 13 amino acids, and in protein C and gene C, it only makes actually 9. Protein C, it's a different form of the protein. And this is what's one of the amazing things. Originally, I'm going to kind of go off on a little sideshow here for a second. Scientists were able to start, well, you remember the Human Genome Project. A whole bunch of different labs were working on it all over the world. They'd be working on different sections. And there's something like, a couple of 100,000 different proteins that, have been found in a human body. And it was thought at that time that you would have like one gene for one protein. Well, lo and behold, there were only 20,000 genes. And yet there were all these different proteins. Well, it turns out that the same gene can make different versions of proteins. It's called alternate splicing. So in a eukaryote in the genome, the gene actually has what's called an intron and an exon. The exon is what codes for the protein. The intron are like spacers. And they actually, in the mRNA, they have to be spliced out. Well, you can make a different form of a protein, let's say protein C, by only putting together some of the exons that actually code in the mRNA, that actually code for those amino acids, and not others. So you can end up with one gene making many, many different kinds of, well, they're called isoforms of proteins. There's another thing. A lot of these genes are highly conserved. The same genes that are involved in making proteins for our eye are also found in Drosophila, which are fruit flies. Exact same genes. There's a few differences, obviously, but it's basically a homologue, or a gene that It's similar because of shared ancestry. Now, each gene has a promoter region that regulates gene expression, and that promoter region is generally upstream of where the gene that you want made is. Okay, now I want to show you something. It used to be thought that a gene was either on or off. The gene was turned on, it made product, that means gene product, which is a protein, or it was turned off and it didn't. It's really not that way at all. In fact, it's more, a better analogy is a rheostat right here. And you can dial it up or dial it down. Okay? You dial it up, you actually make more of the protein product. You dial it back, you make less. Just a dimmer is an example of a rheostat. So you want more light, you dial it up. You want it less light, you dial it back. That's the same way it works. Now that's actually controlled in the promoter region upstream. You have these upstream sequences that actually regulate how much of the gene product they actually regulate, like the rheostat or the dimmer. Okay, so it's not an on or off switch. It doesn't work that way. It's a continuous function of more current or less current, or more gene product or less gene product. Okay, now, it turns out that with humans, a strange thing happened. There were a lot of human cultures that were called pastoralists. So they had animals like cows and goats and sheep, and they were nomads, and so they'd move their herds to new places to feed on new grass. There's still pastoralists today. If you go into Africa, the Maasai are pastoralists, quite a number of different groups. Okay, so in the Middle East, there's pastoralists. In fact, that looks like where it first, pastoralists or that kind of animal husbandry was first kind of perfected. Well, as you can imagine, an adult cannot drink the milk made from, let's say, a cow. Okay, and so those first human pastoralists they didn't have cattle for milk. They had cattle for meat. Some of them also had cattle for blood. What they would do is they'd bleed the cattle, they'd collect the milk, the blood, and they'd drink it. The Maasai still do that. They actually mix it, though, with milk. So, sounds kind of awful, but if you brought up in it, you might actually like it. So, there was a mutation in one individual. That the mutation was that it didn't turn off the lactase gene, and so that adult could actually still digest milk. Now, it wasn't necessarily on or off, but that adult could digest some milk, not as efficiently, let's say, as later on as the promoter region got better. And remember, it's more like a rheostat. Okay, I'll stop this. Okay, so this mutation that allowed adults to actually drink milk and dairy products was a beneficial one, because all of a sudden you had this food source that before you couldn't use, and now you could. And it was an adaptation in this case, because here they had these ungulates that were producing milk, and they could actually use the milk as food. And so individuals that had that, who could actually digest milk and get nutrition from it, obviously did better than those that couldn't, especially when there was a famine for other things. So they left more offspring. It turns out, though, that this mutation actually originated at least 3 or 4 different times in different human populations. There's a population in Northern Europe that's famous, Denmark and Sweden for dairying. The Middle East also had dairying culture and pastoralists. And then there were at least 2 different populations in Africa that were pastoralists that had animals. And in each of those cases, there was a mutation in the promoter region that kept the lactase gene on, and actually, made it so that it made more lactase when it needed it, and less lactase when there wasn't as much milk there. All that kind of stuff. So it was actually like a dimmer. The thing is, it was not the same exact mutation in this stretch of DNA upstream. It was a different mutation, but the mutation had the same effect on producing lactase. Pretty cool. Okay, on producing lactase into adulthood, all the way through adulthood. And those individuals, of course, that had that mutation were able to use lactase and use it to their benefit. There's no more lactose in the system, and the promoter region that turns on the lactase gene is permanently actually shut off. Okay. All well and good. So here we are again. Remember, transcription occurs in the nucleus. You have what's called RNA polymerase that binds to the promoter region of the gene and synthesizes a transcript of the gene. Okay, so you have this RNA polymerase, which is, this green thing right here, the big protein. Anything with an erase or ACE in it means it's an enzyme. So it's catalyzing a reaction. The promoters have distinct sequences that the RNA polymerase recognizes, and it transcribes that. And here we have a whole bunch of RNA nucleotides that are being made from the DNA and binded to it with the RNA polymerase, and then you end up with the mRNA, which is then, of course, taken or ferried out of the nucleus and into the cytoplasm, where the ribosomes then turn it into a gene. Okay. Now, obviously, you also have to make RNA polymerase, so there's also a gene for RNA polymerase. There's usually an RNA polymerase in all cells, because this is kind of a basic cellular function. Making copies of themselves, making proteins, having to make RNA polymerase in every cell so the polymerase can do all this stuff. So in every cell, RNA polymerase is made. So here's the central dogma, and I mentioned before, the central dogma is you can go from DNA to RNA to a protein, but not the other way around. So you have a polymer of nucleotides, 2 complementary strands of of DNA. You then go to RNA. Let's see, G. Okay, so wherever you have a T, you have a U, a T, a U, T, T, U, U. And then now hold on a second. So let's see. Yeah, no, that's right. Exactly. Okay. Hold on 1 second. Okay. So you have this DNA. It's then you got a transcript of RNA, which is the same as this second strand here, right? So it's GCA. Wherever there was a T, it's now a U. Okay. Now, each 3 base pairs codes for a specific amino acid. So the RNA is actually a single strand of nucleotides instead of DNA, which remember is a double strand. And then these different amino acids, a polymer of amino acids, that means a polymer of anything is many of those things. So each nucleotide, which is a base pair, right? You have 3 nucleotides that then code for an amino acid. And then they're strung together in a specific order and you get a protein. You cannot go the other way around. You can't go back from a protein and then translate it into a transcript and then transcript it into a DNA. It doesn't work that way. Okay. Now, translation occurs in the cytoplasm. So, remember, here's our mRNA, or actually, let's see, where are we going here? Okay, we're going this way. So, here's our mRNA. Here's the ribosome. And then what's happening is the mRNA, each 3 base pairs, translates into a polypeptide amino acid. And they're all strung together, and then out comes this protein. And then at the very end, you get what's called a stop codon, and it stops the translation. So translation occurs on a multi-protein complex called the ribosome. The ribosome moves along the mRNA, which is this right here, right? To receive the next tRNA. So, let's see. Yeah, that's right. Okay, so the next tRNA. So, you have the mRNA, and then it's transcribed or translated into tRNA and then amino acids. So the ribosome moves along the mRNA to receive the next tRNA. And then when the ribosome reaches the stop codon, no tRNA can base pair with the codon on the mRNA, and the protein is synthesized and released. So, transfer RNA. The tRNA carries amino acids and matches them with the correct codon. That's the 3-letter mRNA, right? So, here's our codon, mRNA. This is our tRNA. This is the anticodon. So if you have a U, the opposite of that is an A. So remember, a U in RNA is a T in DNA. Okay, so it's this region of internal complementarity. They string together, and this is the binding site for amino acids. And then this is phenylalanine or something like that. I'm not exactly sure. tRNAs, or transfer RNAs, are the interpreters of the actual genetic code. So, it actually takes the code and translates it then into an actual amino acid. Now, this is genetic code. There are 20 different amino acids. Okay. We'll say first base, second base, and third base. Remember? So the first base is in red, the second base is in blue, and the third base is in green. Okay. This is the universal genetic code. It's the same in all organisms. The guy who actually worked this out was one of the two who discovered the structure of DNA, and that was Francis Crick. So he also figured all this out, and then later in life, he switched completely and went into studying the brain and neural networks and things like that. Brilliant guy, absolutely brilliant. But anyway, when you change a first base pair, you will automatically change the amino acid. So, okay? So, if you go to a U to a C, you'll go from either a phenylalanine or a leucine to a leucine. Or, if you went to an A, you would go to an isoleucine. Or if you went to a G, you would get to a valine. Okay? So each changes in the first base pair of the codon, you'll get a change in amino acid. Now, if you have a change in the second base from, let's say, a C, let's see, hold on a second. Okay. Oh, here we go. We're going across this way. So you go from a U to a C, you get serine. If you went from a C to an A, you would get either tyrosine or a stop codon. And then if you went from, let's say, an A to a G, you get cysteine or a stop codon or tryptophan. Now, so each time you make a change in the base pair of either the first base pair or the second base pair, you get a different amino acid. But the third base is different. Third base is often what's called degenerative, either 2 times degenerative or 4 times. So in some cases, if the third base is, if you change it, let's say, from a U to a C, you'll still get an arginine. If you'll change it to an A, you'll still get an arginine. If you turn it from an A, let's say, into a G, you'll get an arginine, as long as these two first ones don't change. So in this case, the C and the G, they don't change, but the third base pair, which is the green, okay, if the green changes, a lot of times you get the same amino acid. When it's 2 times degenerate, like here, you'll get either a serine, if it's a U or a C, or arginine, if it's an A and a G in the third base pair position. For glycine, it's like arginine. So if you have a G, G, and then a U, C, A, or G, you'll get a glycine. If you have a CG, and then a U, C, A, or G, you'll get an arginine. When it's 2 times degenerate, you'll either get a cysteine or a stop codon, UGA, or a tryptophan UGG. Okay, so universal genetic code. So how does DNA determine our traits? DNA doesn't determine our traits. Proteins determine our traits. So you have DNA that codes for a certain amino acid sequence, which is then, that's the actual protein. And remember, we go from DNA bases to an mRNA intermediate called, it's an mRNA transcript that's ferried out, and then that's strung together in a ribosome into transfer RNA and then to amino acid, where it adds amino acids. And so the actual proteins are the ones that do the work. In this case, taste receptors. We've got proteins that recognize taste receptors. We've got, for bitter, we've got taste receptors for sour, salty and sweet, sour, and right in here, not many taste buds. There's also another kind of central taste. It's called savory, and it's found in many meats. So, the hemoglobin gene. This is what carries our blood. These are red blood cells. The hemoglobin gene carries oxygen. It picks up the oxygen in the lungs, and that's ferried to the rest of the cells, where it gives up the oxygen and then takes in CO2, carbon dioxide. Takes that out. That's then transported out back to the lungs, and then it's on our out breath, it's out into the air, and we take in another breath. Okay, now, there's actually the hemoglobin gene, it's on chromosome 11, but it actually comes in two different forms. They're isoforms of the same one. One of them can carry 4 oxygens, the other can carry 2. So at rest, you're going to have a population of hemoglobin that's going to be mostly the kind that carries 2 oxygens. As you start to ramp up your, let's say, exercise, or you're starting to burn up fuel because you're, you're running or something like that, then most of the hemoglobin, what will be produced is this hemoglobin that will carry 4 oxygens at the same time instead of 2. So more oxygen to the cells. The same thing sort of happens if you go up in altitude. At sea level, you'll have, at rest, you'll have the 2 oxygen hemoglobin. If you go up to, let's say, 15,000 feet, you'll be short of breath, and almost all of the hemoglobin will be carrying 4 oxygens at a time to make up for it. Okay, so it turns out that's as far as we actually could have stopped earlier. So I just wanted to kind of make my own little like Echo 360, the lecture capture, because we didn't get that recorded. So hopefully you can follow along with that. Okay, and then I'm going to go to the very, very end, because we didn't quite finish up, and we're going to have our next test next week. So here we go. I'm going to go to the bitter end. Now, sometimes we have a hemoglobin, normal adult hemoglobin, that has a mutation, and that mutation actually causes sickle cell disease. So, I'll take the blue. Okay. So, normal red blood cells, this is a sickle cell. Does not carry oxygen very well. So, this is the mutant hemoglobin gene. Now, if you only have one copy of it, then you have a red blood cell that will carry oxygen, not quite as well as a regular blood cell with normal hemoglobin, but it will also be a red blood cell that will be resistant to the malaria parasite. So, if you have one copy and you live in an area where there's malaria, you will be resistant to the malaria. You won't get as sick, or you won't die. But, unfortunately, if you get two copies, one from your mother, one from your father, then you have what's called sickle cell anemia, which is a recessive deleterious mutation. Individuals with sickle cell, they rarely, well, more of them are now, but they rarely live to adulthood, and they rarely reproduce. So in other words, this is actually an adaptation to fight malaria, but there's a pretty high cost because all individuals with the mutated form, both from the mother and the father, they all die. They don't leave any offspring. Okay, hold on. Okay, so this is kind of where we left off. We were talking about introducing DNA with the gene gun into a plant, into a plant cell. We also can do the same sort of thing with a gene gun that can be used to put DNA into humans. This is called what is it? DNA therapy or oh God, I don't know. Anyway, it's just starting to be used again successfully. When new genes were first introduced trying to correct a genetic disease where they had the wrong mutated form of a gene and they tried to introduce the right one. It seemed to help, but then a lot of the people who had it done eventually produced tumors and died of cancer. So that went on hold for a long time until now that we understand things a little bit better. Okay. So you've made a transgenic plant. You finally created a transgenic, you've kind of made a transgenic. Okay, you finally created a transgenic corn plant that is resistant to pests. You expand the strain and sell the seeds, to 95% of the corn farms in the U.S. Lots of dollars. What do you think you've done to the diversity of corn in the USA? You finished everything. You end up with clones. All the same corn, exactly clones of themselves. No diversity at all. Now, you may say, well, but this corn is so good, there's such a high yield that so what? But if you had a pathogen that came along, let's say it was a bacteria or a virus or a fungus or something like that, since there's no diversity, probably every plant with the same clone genome would be susceptible and they would all die. So, even with selective breeding, the same thing happens, but especially with like transgenic clones. And so, right now, most of the corn that we grow is actually clones. Exactly the same corn, same genome, same exact corn plants. And if a pest ever comes along that this variety is susceptible to, it'll all die. In fact, the exact same thing is actually going on right now with our banana. So I already talked about this, but this is pretty much, I think we're at the very end. Okay, bye.